Welcome to NephJC, a Twitter-based nephrology journal club for physicians, students, and patients. My name is Jenny Lin, and I am a physician scientist and attending nephrologist at Northwestern University's Feinberg School of Medicine. In this video, I will be giving you a recap of the exciting NephJC chats from December 11th and 12th, 2018. Up for discussion were two recently published papers that have launched kidney science into the high-impact spotlight. These studies use single-cell RNA sequencing technology to highlight the beauty and complexity of the kidney. The first study, entitled "Single-Cell Transcriptomics of the Mouse Kidney Reveals Potential Cellular Targets of Kidney Disease," was published by Dr. Callan Shustak's group in the journal Science in April 2018. This study was among the first to characterize the mouse kidney on a single-cell RNA level and demonstrates the power of single-cell sequencing technology in discovering novel cell phenotypes. Dr. Shustak is no stranger to Twitter and actually won the NephJC Engaged Scientist of the Year award in 2017. She has also been so excited about single-cell sequencing that she actually decorated her famous cakes with T-SNE plots. We will review T-SNE later. The second study, entitled "Comparative Analysis and Refinement of Human Pluripotent Stem Cell-Derived Kidney Organoid Differentiation with Single-Cell Transcriptomics," was published by Dr. Ben Humphrey's group in Cell Stem Cell in November 2018. It has gained international attention for its careful single-cell RNA characterization of stem cell-derived kidney organoids grown in culture. If you don't know what a kidney organoid is, that is okay. Just think of it as a 3D cell culture model for kidney cells that consist of immature podocytes and tubular-like cells and structures. They grow from pluripotent stem cells that have the potential to differentiate into whatever you direct them to become. Dr. Humphreys is also quite a prolific tweeter and, like Dr. Shustak, integrates single-cell biology with different aspects of his life. Before we dive into these exciting papers, we will briefly review some background on why we would do single-cell RNA-seq and how it works. For more information, you can also read my summary on the NephJC website. So, prior to single-cell methods, we would perform RNA profiling by way of bulk RNA-seq or microarray on tissues that have been lysed for RNA isolation. We have seen some interesting and valuable data from this approach. However, as we all know, the kidney consists of many different cell types that work in coordination with each other to perform complex physiological tasks. Mixing together RNA from all parts of the nephron doesn't quite do the kidney justice. Researchers have tried to circumvent this through microdissection of different structural components of the nephron, separating out the glomerular compartment from the tubular compartment. However, microdissected kidney samples still contain multiple cell types, so we wouldn't necessarily know whether an RNA signal truly is from the podocyte or endothelial cells within the glomerulus. To get a higher resolution picture of what is going on in a complex organ such as the kidney, scientists have been turning to single-cell RNA-seq. There are several platforms for making single-cell sequencing libraries, but I will talk about the droplet-based method used in both papers we discussed, and more specifically the 10x chromium method used in the Shustak paper. The process of single-cell RNA-seq can be summarized in the following steps. Step 1. Make a single-cell suspension from a sample. A tissue or organoid sample needs to be broken down to its single-cell components to make a suspension of living, intact single cells. This is perhaps one of the most challenging aspects of the workflow, because complex tissues such as the kidney can have more sensitive cell types such as podocytes, which are more likely to die during the dissociation process and thus not get represented in the sequencing. We try to target over 80% viability in a sample to reduce capture of RNA from neighboring dead cells that don't represent your cells of interest. In some tissue samples, like say a fibrotic kidney, this is especially difficult, so you might opt to isolate single nuclei instead. During the chat, nuclear preps were briefly discussed by Dr. Humphreys and Dr. Shustak, who said that with nuclear samples, you can get more cell types represented with the added advantage of not wasting sequencing reads on mitochondrial RNA, although not all cellular transcripts will be captured. During the chat, Dr. Sparks also wondered about potential biases in the single-cell suspension step, and Dr. Nelson asked about how we might decrease representation from the proximal tubule so that more reads go to the other cell types. Dr. Chu countered with the question of introducing more bias by depleting cell types. Dr. Humphreys acknowledged that methods for dissociation can alter relative cell population abundance, and it's unknown how much bias this introduces. Step 2. Give each cell a unique ID code that gets added to its transcriptome. Each cell needs to be marked for identification purposes so that data from a single cell can later be identified as originating from that specific cell. Why are we not making a different library for each cell one by one and then sequencing each cell one by one so that we can keep track? Well, this would be an impossible task in terms of time. If you think about it, it's not practical to make a separate, isolated microlibrary from each cell one by one sequentially, since the workflow takes multiple hours per library, and imagine multiplying that by 4,000 cells. It would take you more than 3 years to make 4,000 separate, expensive libraries if you made them in a series instead of in parallel. Now, to make single-cell sequencing more efficient and scalable, we pool together or multiplex all of the molecular materials from all the cells of a sample together into what we call a library. The data in the library can be read simultaneously, and this is the key to making the process feasible from a time and cost standpoint. That's why we do everything in parallel in a pooled fashion. To figure out what belongs to which cell, we give each transcript an ID barcode that corresponds to the parent cell. We then leverage computer technology that allows us to demultiplex or organize the data and IDs on the back end. The general concept of multiplexing and sequencing in parallel is what got us out of the low throughput methods like Sanger sequencing into next generation sequencing. So again, when all the RNA from all the cells are pooled together for massively parallel sequencing, we have to have a method for identifying which RNA transcripts come from which cell in order to see our data in a cell-specific manner. Does that make sense? In my write-up for the chat, I used lucky podocyte number 13 as an example. To perform single-cell sequencing, we need to be able to identify which transcripts are from podocyte number 13, so all of the RNA from podocyte number 13 needs to have an ID barcode telling us they belong to podocyte number 13. This means that later we can find all of the NPHS1, also known as nephrin, transcripts from podocyte number 13. So to recap, all transcripts from podocyte number 13 are labeled with a barcode that tells us they are from podocyte number 13. Okay, so now each transcript also has a unique molecular identifier, or UMI, that goes into the sequencing recounts. This is separate from the cell ID barcode. We would expect there to be more than one copy of NPHS1 in podocyte number 13 because nephrin is very important in podocytes, and each NPHS1 RNA molecule gets a different UMI but the same podocyte number 13 ID. The UMI system allows us to count how many unique NPHS1 transcripts there are assigned to podocyte number 13. It also allows us to identify sequences that are PCR duplicates from library amplification, or in other words, technical artifacts, rather than unique transcripts representative of the biology of the cell. So, taking a step back, once a single cell suspension is made with greater than 80% viability across cells, the cells enter a microfluidic system where each single cell is co-injected with a gel bead into an oil droplet emulsion. Each gel bead contains a different barcode to identify the cell, as well as UMIs to identify transcripts, the combination of which marks a single cell. The RNA is reverse transcribed into complementary DNA or cDNA, incorporating the barcodes and UMIs in the process. Step 3. Make cDNA libraries from the droplets containing single-cell cDNA material. To do this, the droplets with the beads are lysed, the cDNA is cleaned up, and the cDNA is then amplified by PCR into a library of sequences to read. Now, remember I mentioned that we sequence everything together? This same principle applies to sequencing multiple libraries, which we pool together to minimize sequencing batch effects. So, to demultiplex the libraries later, each replicate sample is also given a sequence index barcode so that we can identify which transcripts belong to a particular replicate. Step 4. Sequence the libraries. This usually happens at an academic or company core facility on a high-throughput sequencer such as an Illumina HiSeq or NovaSeq machine. Step 5. Demultiplex and analyze the data. After the sequencing run is complete, the core facility will release your data to you. The files are processed so that you can extract and sort out reads from each library. This process is known as demultiplexing. After demultiplexing is complete, computational efforts then generate a differential gene expression matrix. No, not that type of matrix. This type of matrix. A series of quality control steps are then taken to minimize technical artifacts and low-quality reads. These steps are crucial to understand so that you can tweak as appropriate for your samples and your experimental conditions. For example, for most single-cell data, investigators like to eliminate cells with high mitochondrial read content that may be more indicative of cell death that increases access to mitochondrial RNA. However, as nephrologists, we are interested in kidney cell types, and mature proximal tubular cells are high-energy utilizers and therefore would be expected to have high mitochondrial reads. So filtering out those cells would potentially reduce the number of healthy cells you would later classify as being part of the proximal tubule. Okay, so once the filtered reads are ready to go, you'll see that the data are very multidimensional because you are comparing tens of thousands of genes expressed in one cell to the same large number of genes expressed in a different cell, but in a different pattern. Most analytical methods reduce the high level of multidimensional data into fewer dimensions that can be more easily visualized and interpreted. And principal component analysis, a linear dimensionality reduction method, is a popular way to do this, at least at first pass. T-distributed stochastic neighbor embedding, or T-SNE, is a non-linear dimensionality reduction method that allows you to visualize high-dimensional data sets. What does that mean? The similarity of one cell to all other cells is first calculated into a similarity score based on the transcripts. Cells that are more similar to each other end up moving closer to each other on a plot, while those that are more different end up repelling and moving away from each other. This captures local relationships, and these relationships are always non-linear in structure. This also creates a probability distribution of similarity scores that are Gaussian in distribution. When plotting these points, you end up with many points concentrated towards the middle because that is how Gaussian distribution tends to be. Now, when recreating the data in low-dimensional space in a student T-distribution, the crowding problem in the middle is improved, and thus embedding of visualized data points are more optimized. In other words, you can see the clusters of cells better and they're not all concentrated in the same place in the middle. There are other types of analyses that can be performed with your single-cell sequencing data, but this should hopefully be a good starting place for getting familiar with the paper selected for LeftJC discussion. Okay, so let's get back to the literature. The first paper on deck for discussion is a science paper from Catalin Shustek's group. They characterized around 57,000 single cells from healthy mouse kidneys and found a novel cell type in the distal tubule. During the chat, Dr. Shustek said that she was inspired to execute this project to figure out which cell types in the kidney she would want to carry forward to study in the future. The second paper is a cell stem cell study from Ben Humphrey's group. They compared kidney organoid differentiation protocols by sequencing around 83,000 cells from 65 organoids differentiated from pluripotent stem cells. They compare these transcriptomes to that of the human fetal kidney and the human adult kidney. Dr. Humphrey stated in the NEF-JC chats that his group pursued this project to evaluate the fidelity of organoids to the human kidneys and what off-target populations may form within the organoids. Through their analysis, they found a pathway to target to reduce the development of non-kidney cell types in the organoid model system. Both papers start off showing the single-cell transcriptomic profiles of the samples sequenced. These profiles are more quantitative than what we would see on histology, for example. In the Shustak paper, we see some of the usual suspects we hope to find on the T/E plot in Figure 1. Every dot represents a cell, and this is how the cells cluster by how similar they are to each other. You'll notice that cluster 2 represents podocytes and cluster 3 represents proximal tubule, which can be further subclustered there on the left. These clusters are determined by relative enrichment of marker genes, which you can see down in the corresponding violin plot. The violin plot shows a visualization of how many cells from each cluster express each marker. For podocytes in cluster 2, we would expect enrichment for NPHS1 or nephrin and NPHS2 or podocin. You can see these marker genes are not highly expressed elsewhere. Notice in the violin plot, and cluster 8 includes markers for both principal and intercalated cells. We will come back to this later. The Humphreys paper also presents the visualized clusters of the organoids they sequenced in these T/E plots. Here I am showing you Figure 2. M denotes the Morizane protocol and T denotes the Takasato protocol for kidney organoid differentiations. You'll see that there are podocytes in differing relative abundance, proximal and distal tubules, and even neurons and muscle cells. We will come back to these off-target cells later. The marker genes here are presented as heat maps, although violin plots of similar T/E plots are presented in supplemental Figure 1. In the heat map, dark bands mark the expression of marker genes in relevant clusters. Sapotic cell and NPHS2 mark podocytes and are not really expressed elsewhere. Next, both papers tackle the connection between cell type and genes associated with human disease. In the Shustak paper, Figure 2 shows where disease-associated genes are expressed in the mouse kidney. Looking at these heat maps, you can see that most genes are cell type specific. For example, in nephrotic syndrome, you can see that these genes are primarily in the podocyte, a straight line down. Renal tubular acidosis is not surprisingly enriched for genes expressed in the intercalated cells of the collecting duct. We also see predominantly one cell type expressing genes found in CKD genome-wide association studies, or GWAS, which identified novel genes associated with complex traits such as CKD. Interestingly, we see a similar pattern in Figure 5 of the Humphreys paper, which showed mapping of expression of GWAS hits to a human adult kidney biopsy his group sequenced on the single-cell level. However, the organoids did not express many of these genes. This is likely due to the relative immaturity of the organoids, which don't completely overlap in terms of cluster expression patterns with the adult kidney, seen in pink, on the overlapping T/E plots in Figure 4. This is further broken down in looking at mature podocyte and tubular markers. You'll notice that the dark red squares are expression of marker genes in the adult kidney. These markers include nephrin for the podocyte and megalin and cubolin for the proximal tubule. There is faint nephrin expression in the organoids and no expression of megalin or cubolin. However, there are developmental markers expressed in the organoids and none in the adult kidney. Finally, these cutting-edge efforts to see the kidney in single-cell resolution paid off for both groups, as they ended up reporting novel findings. In the Shustak paper, remember I pointed out that cluster 8 included markers for both principal and intercalated cells? The Shustak group decided to explore this cell type further as it may have implications for acid-base handling in human disease. They stained for principal cell and intercalated cell markers and found in lower abundance the presence of cells that stained positive for both, validating the single-cell RNA-seq data. A different type of single-cell RNA-seq analysis called pseudotemporal ordering, or pseudotime, which is performed by a program called Monocle, can show how similar cells are to each other based on similarity of gene expression profiles, but on an axis of time. Using traditional principal cell and intercalated cell markers for branch points, the Shustak group found that the novel cell type in blue indeed sits between the two cell types in terms of similarity and is thus a transitional cell type. This is also confirmed by lineage tracing experiments in the mouse, where you have GFP expressed in cells with an ATP6A lineage tag. You can see that some cells stain for 3 colors, the GFP tag, and a marker from each cell type. These cells are thus the transitional cell type. In examining gene expression differences between principal cells and intercalated cells, the Notch signaling pathway came up with differences in ligand and receptor expression according to cell type, as seen in the heat map in Figure 4. Turning on the expression of a portion of Notch in adult mouse kidney shifted collecting duct cell patterning with increased principal cell markers. This was also seen in the folic acid model of renal fibrosis in mice, which was tested due to the known involvement of Notch in renal fibrosis and CKD. Consistent with the change in collecting duct phenotype and renal tubular acidosis, serum bicarbonate was not detected in the folic acid mice. Taken together, the intercalated cell to principal cell transition may be mediated by the Notch pathway, and a shift towards the principal cell fate may drive metabolic acidosis seen in renal fibrosis of mice and potentially CKD patients. The Humphreys group leveraged their single-cell data to optimize organoid differentiation protocols. Remember I pointed out that they found muscle and neuron cells in their organoids? This made big news. In their pseudotemporal ordering analysis, they found a branching point between neurono-mesenchymal cell fate and podocyte cell fate. They performed a finer receptor ligand analysis between the podocyte and neurono-mesenchymal clusters. They found 19 receptors and cognate ligands, and NTRK2 was expressed exclusively in neuronal clusters. Its ligand, BDNF, was expressed in the podocyte neuronal branch. Because BDNF promotes neuronal survival and differentiation, the authors hypothesized that inhibiting its signal would decrease the growth or formation of neuronal clusters in the organoid differentiation process. They administered an NTRK2 inhibitor to growing organoids and found a 90% reduction of neuronal cells from about 20% down to 2.1%, and an increase in proximal tubular cells and podocytes. This experiment shows that this approach and use of single-cell RNA-seq can be leveraged to refine and optimize organoid differentiation protocols to reduce off-target cell types and enrich for the types of cells we care about. Of course, with any new technology, there are technical challenges and limitations. These were not discussed extensively during the NFJC chat. One major limitation is that we get relatively low coverage of the transcriptome with droplet-based sequencing methods, and certainly analyses for alternative mRNA splicing would be missing quite a bit of data. There is also quite a bit of variability from cell to cell, with more noise than we typically see for bulk RNA-seq. Also, the currently available analytical methods are not as mature and well validated as they are for bulk RNA-seq, so quality control of the accepted methods is needed. Even with caveats, single-cell RNA sequencing has already proven to be a powerful tool to aid investigators in making important discoveries in kidney science and beyond. Here are some comments from the NFJC participants on how they view single-cell. If you would like to read more in-depth reviews of these papers, please check out the following pieces from Dr. Andy McMahon, developmental biologist, and AJKD, and from Dr. Bino Friedman, an organoid expert who has also used single-cell RNA sequencing in his high-impact work. His piece can be found in the journal Nephron. Thank you for tuning in. We hope to see you online at future Basic Science NEFJC chats. Tweet you then!